Hello everyone, and once again I welcome you on this new topic on respiration, which is also very important for your NEET examination. Now, as per my experience, the most difficult topic, or the topic which is not understood by most of the students, is electron transport system or terminal oxidation. So that is why I thought of throwing some light on this topic, which will help you to understand it much better. So let us see what you mean by electron transport system or terminal oxidation. The conversion of the reduced coenzyme into their oxidized form is called as terminal oxidation. Now, NAD is a coenzyme, nicotine adenine dinucleotide. This is the coenzyme. NAD is the oxidized form. When NAD is with H2, when it accepts H2, it gets reduced. Now, conversion of this NADH2 into NAD is called as terminal oxidation. So, NADH2 is oxidized and NAD is formed. So, when oxidation takes place, hydrogen is removed. So, this removal of hydrogen is then accepted by FMN. This is flavin mononucleotide. Okay, and then the series goes on. So first, let us try to understand terminal oxidation. So this terminal oxidation involves the electron transport system. So once again, the conversion of the reduced coenzyme into their oxidized form is called as terminal oxidation. And this terminal oxidation involves the electron transport system. The metabolic pathway through which the electrons pass from one carrier to another is called as the electron transport system. Now, where do these electrons come from? When hydrogen is removed, at the same time, hydrogen is accompanied by electrons. These are called as the hydride electrons. Hydrogen is nothing but the proton, and protons are always accompanied by electrons. So, when hydrogen is removed from any substance, or when oxidation takes place, hydrogen is removed, and along with hydrogen ions, you have hydride electrons also. So, these protons as well as electrons are accepted by FMN. This is flavin mononucleotide. When after accepting hydrogen, flavin mononucleotide gets converted into FMNH2. Now this FMNH2 transfers the electrons as well as the protons to coenzyme quinone. Now this coenzyme quinone, it accepts the electrons as well as the protons. The protons are transferred into the mitochondrial matrix, while the electrons are transferred to a series of electron acceptors. So let's see how the electrons are transported. The electrons are first transported to cytochrome B. From cytochrome B, they are transferred to cytochrome C1. From here, they are transferred to cytochrome C, then cytochrome A, A3. And by the time they reach cytochrome A3, the energy of these electrons is lost and they become de-energized. So when these de-energized electrons, they are accepted by oxygen, oxygen gets activated. And when the oxygen gets activated, it combines with the hydrogen or the hydrogen ions from the mitochondrial matrix and forms a water molecule. So this is the way the electrons are transferred and the water molecule is formed. So this process occurs in the inner mitochondrial membrane, that is the cristae. So this is all about the terminal oxidation. So electron transport system and terminal oxidation, they go hand in hand. Okay, to explain terminal oxidation, you need to explain electron transport system, and to explain ETS, you need to explain terminal oxidation also. So this is in brief about terminal oxidation. Now we'll just go through some frequently asked questions. So the first one is, in eukaryotes, photosynthesis takes place in chloroplast and respiration takes place in the cytoplasm and mitochondria. Then carbohydrates are the most perfect substrate for biological oxidation. The third point which you need to know is plants do not have any specialized organs for gaseous exchange. They only have the stomata and the lenticels. Now, what is the reason that plants do not have respiratory organs? Now, the main reason behind this is that there is very little transport of gases from one plant part to the other. Then the second reason is that the root, stem and leaves respire at a very low rate as compared to animals, and that is why they do not need any respiratory organs. And the third reason is that most cells of a plant organ have at least one part of their surface in contact with the air, and therefore diffusion takes place very easily. So because of all these reasons, they do not have special respiratory organs. Instead, they have only stomata and lenticels through which the exchange of gases takes place. Now the next point is the types of respiration on the basis of respiratory substrate. Now on the basis of respiratory substrate, there are two types of respiration: floating respiration and protoplasmic respiration. Now when the respiratory substrate is fats or carbohydrates, the respiration is known as floating respiration, and when the respiratory substrate is a protein, it is known as protoplasmic respiration. So these two types of respirations you should know: floating respiration and protoplasmic respiration. Then glycolysis. Glyco means sugar and lysis means splitting. So the splitting up of sugar is known as glycolysis, and this was discovered by Gustav Emden, Otto Meyerhoff and J. Parnas. So this is also known as EMP pathway. Mostly you know only Emden, Meyerhoff and Parnas, but the complete names are, or the full names are, Gustav Emden, Otto Meyerhoff and J. Parnas. Then the next point is Gay-Lussac first discovered fermentation. Again here you should remember the name of the scientist who discovered fermentation, that is Gay-Lussac. The next point, maximum concentration of alcohol in beverages that are naturally fermented is 13%. PPP, Pentose phosphate pathway, or HMS, hexose monophosphate shunt, occurs in cytoplasm and chloroplast in the presence of oxygen. It is found in both eukaryotes and prokaryotes. It is also called as Warburg-Lipman-Dickens pathway. This pathway is important in tissues like liver, adipose tissue, germinating seeds, ovary, etc. It acts as a safety valve or shunt to EMP. The next point, plants growing in the cold region show cyanide resistant alternative pathway. Cyanide resistant alternative pathway which participates in the oxygen uptake. They oxidize NADH2 and consume the released heat. Now as I told you before, when NADH2 is oxidized, that is when hydrogen is removed, energy is released, and that energy is used by the crops or the plants growing in the temperate regions. Temperate region means a region which has low temperatures. Then 2,4-dinitrophenol, 2,4-DNP, inhibits ATP synthesis. 2,4-dinitrophenol inhibits ATP synthesis. Cytochromes are Fe-rich proteins discovered by Mac Kuhn. Cytochrome A3 has both Cu and Fe and is a component of cytochrome oxidase. So this is an important point. Cytochrome A3 has both copper as well as iron and is a component of the cytochrome oxidase. Another important point about ATP which all of you should know, that ATP is mobile inside the cell, and this is the reason why it can carry the energy away from the site of respiration. 8.15 kilocalories or 34 kilocalories of energy is liberated upon hydrolysis of one molecule of ATP into ADP.